Okay, so the first thing that I want to say is I'm sorry, but I did not get the learning objectives posted for exam number one. I will get those up by the end of this evening. Um, so look out for those. Our first exam is going to be on, it's 2 weeks from today. That exam's going to be administered on Proctorio. Um, in addition to that, we've had some quizzes. I've gotten some emails. No, wait, no, I haven't had. I'm sorry. Sorry, I got y'all mixed up with a different class. I'm going to be posting a quiz today, and that will be open. Let's see, where's our whiteboard? Nope, not that one. Oh, we can start. No, not that one. Share. I'll go with document. Yeah, we'll go with whiteboard. And I want to share it, but I only, yeah, this is the thing that gets tricky. I don't want anyone else to be able to write on it. So I think what I'll do is I'll just go into the PowerPoint and share that. There will be basically a practice quiz for making sure that everyone's able to use Proctorio. So this will be a Proctorio quiz. And then your first kind of content quiz. Now, I'm going to use the term quiz just because that's what they're going to pop up as on Canvas. But keep in mind, these are, open note document or open note kind of questions, more or less. It is going to be a grade. And so, so everything that goes into all of the different quizzes are going to be graded assignments. So they will populate, they will be a part of your grade. They're open note though, so go ahead and use your notes. In addition to that, you're going to have 2 attempts for any content quizzes. So make sure that you take advantage of that. You're not going to get feedback on, or you'll be able to see the answers that you submitted and you'll get a score, but you won't be told what which ones were right and which ones were wrong. So that's the part where you kind of have to reflect and analyze it. So expect those to be posted today, and those will be up posted. That'll be 9/9. And those will remain open. They will close on the 11th at 11 p.m. I was going to do 48 hours, but what I'm going to do is basically adjust that a little bit so you get a little bit more time. And I found that from my other classes, it's easier whenever they are open throughout the entirety of the day. So these will be posted later today. Essentially, they will be a way for you to figure out, are you using Proctorio properly and everything like that? Are you good on that? So before I proceed, does anyone have any questions? You can add it to the chat or say it aloud. I've got volume up a little enough. Okay. Well then, what we are going to get into today is maybe one of my favorite things to talk about, and I'm really excited to share with y'all glycolysis. We talked about it a little bit. I gave you a little bit of a preview last week, but here we're getting into the pathway as a whole, and we're going to break it down over today and Wednesday. And I think that we'll be able to get after everything that you need to know for this exam or this first exam, and you'll be in a good position to know the different moving parts of it. So with that in mind, I'm excited to talk to y'all about glycolysis. Here it is. This is a chart that I think is really worthwhile for you to kind of process and understand. Now, there's a lot of information on this, and depending on how you operate, how your brain works and everything like that, it might be a little bit easier to get like a piece of paper and cover up, cover this portion of it. Yeah, cover like basically cover 2/3 of the page and then say, okay, I'm going to commit to memory this part. Um, you might be the sort of person who's like, I can process all this information in one motion. I'm not that kind of person. The way that I process this and the way that I kind of worked through this was by starting over here. Starting in this column and trying to figure out different parts of it. Now, the most important part of the pathway known as glycolysis is where you start. Now, where you start is with the molecule glucose. Now, what we talked about last week and previously is that glucose is a 6-carbon aldose. And you could take that information and you could say that it is an aldohexose. Okay, so it as an aldohexose, well, that gives you a little bit of information. Now, if you look at the structure of glucose, I know that whenever I talked about glucose, I referred to it in its linear form. I referenced when glucose is in its linear form, I said right, left, right. Now, whenever I talked about glucose in its ring form, what did I say? And go ahead and add this to the chat, somebody. If I said right, left, right when it was linear, what did I say whenever it was cyclical? Dud, or well done. I like that. Down, up, down. Sorry, I got a separate question. We only have to learn the concept portion or the concept first, then enzyme structure and so on. You will have to know all of this information. You're not going to have to draw anything, obviously, but you'll have to be able to identify sugars and know the enzymes that use those different sugars. So I got this separate question that was, we have to learn the concept portion or the concept, then enzyme structure and so on. That sounds to me like a strategy. That's, I think, an effective way to do it. Get a 10,000 foot view and then zoom in. Get a 5,000 foot view, then zoom in to 1,000 and to 100 and so on. So I hope that's helpful. Now, with that in mind, we've got glucose right here. We talked about this being a ring form and down, up, down. Now, with that said, what if I had the C3 epimer of glucose, or heck, the C4 epimer of glucose? Well, the C4 epimer of glucose is galactose. Now, does galactose get metabolized in this exact same way? It does not. Galactose is not going to be processed in this exact same fashion. So what that means is that if you were to take galactose and kind of input it into the glycolysis machine, you're not going to magically get glucose 6-phosphate or heck, galactose 6-phosphate. And what that also means is that this enzyme is not going to be able to process it. Instead, what you're going to have to do is convert galactose. I'm going to erase this arrow. You have to convert galactose to glucose. So you have this conversion step where you're effectively making the C4 epimer of galactose, which is glucose. So that's one of the first things that I think is important to get clear. Now, what we are going to be talking about today is we're going to be talking about basically this first half up until that step right there, where we create this molecule known as fructose 1,6-bisphosphate. This is known as the investment phase. Can somebody add to the chat why they think that it's known as the investment phase? What are you investing? It needs ATP. We add the ATP. That's absolutely right. Great job, Andrew and Kate. What we're doing is we're adding that ATP. Okay, so here's our first ATP that's being added, and here's our second ATP that's being added. So essentially what that means is I'm going to say -2 ATP, because I like to think about this almost as like accounting in some respects. We've spent 2 ATP, so we're in the hole 2 ATP. It's what's it called? Deficit spending. Then what do you think the other portion of this pathway is referred to as? If the first half is the investment phase, what's the second half? Add it to the chat. And there's not one specific, right? I like it. Payoff, the return, the profit, any one of these terms works perfectly for this. So let's go with payoff was offered up first. So I'm going to go with that. The payoff phase. Okay. So what that means is we're basically offsetting that ATP. So let's highlight those steps where we're getting that ATP back. Well, one step is going to be right here. Now, what's important to recognize about this step is that we're taking 1,3-bisphosphoglycerate and another 1,3-bisphosphoglycerate to make 2 molecules of 3-phosphoglycerate. And I'm telling you these names because we're going to get to them. And at the moment, you might not really have any context, but we're going to get there. We're getting 2 ATP because what we're doing is we're removing 2 phosphates from 2 3-carbon molecules. Because we started with 1 6-carbon molecule, then we're going to get to 2 3-carbon molecules. Here, we're going to get ATP, get 2 ATP, in fact, plus 2 ATP. So now we're back at a total of 0 ATP. So we did our deficit spending. We reaped a profit. We're back at 0. And then this very last step at the bottom. Well, that's another step where we're going to be generating ATP, but we're actually generating 2 ATP. So this point, I'm going to say plus 2 ATP. Because that's the step where we're going to get our ATP back and we're actually going to be profiting. So let me clear this up so that it doesn't look like I said 12 ATP plus 2 ATP. There we go. So this is going to get us to a place where we invest, we get our return, and now we're above. What is it? We're in, I think we're in the black because in the red is bad. So we're in the black. We've gotten 2 ATP back. Now, that's one of the big takeaways from ATP. Okay, is there a reason as to why it says ADP instead of ATP? That's a great question. The reason is because ADP, notice how it says it in both places. Well, let's zoom over a little bit and look here and here respectively. The reason is because we're investing 2 ADP. So 2 ADP are being used in order to get back our 2 ATP. So this ADP is merely like you're adding adenosine diphosphate. You're adding a phosphate to that, and then you're getting ATP. Does that make sense, Summer? Awesome. But I'm happy that you asked that because that's the sort of question that you are not the only person who had that, because if you that's why I think it's important whenever you break this down, this is not a standalone understanding of glycolysis, because if you look through that and you say, oh, okay, 2 ADP, 2 ADP, you're going to think, I get ADP out of this? But only after you've kind of pieced it all together do you see that, yes, indeed, you get ATP, but you're adding 2 ADP, so you get 2 ATP. Okay, so one of the things that I also want to add about that and highlight as your kind of big overview is this statement right here. There are 3 steps that are irreversible. Now, personally, whenever I think of an irreversible step, the first thing that comes to mind to me is a one-way road. You've got one direction that you can go on that road. If you go the other direction, you're going to be in trouble. And I mean, either legally or another car is going to hit you. Now, what I want you to highlight about that and recognize about that is here is one step where we are investing 1 ATP. Here's another step where we're investing a second ATP, so 1 ATP. So both of our steps where we invest ATP are irreversible. Then if we go down to the bottom, we have two steps where we're generating ATP. One of them is this step right here, our first of them, and then our second one is right here. So we have two steps where we invest ATP. Those are both irreversible. Then we have we have two steps where we return ATP. And one of those two steps is irreversible. And we'll talk a little bit about the why a little bit later on when we're on Wednesday when we're dissecting the payoff phase. But I think that's an important thing to remember. Okay, what are my irreversible steps of glycolysis? And why is it important to remember those? Because those are your very tightly regulated steps. When you think about regulation and a process, going after those very tightly regulated steps are kind of the bottlenecks for how well this whole system moves and how easily this system moves. So the other 7 enzymes in this pathway are completely reversible. So I always like to think about this as percentages. 70% of glycolysis is reversible, and 30% is irreversible. Okay, got a question in the chat. Since glycolysis invests only 2 ATP, but returns 4, sorry, I need to correct that, returns 4 ATP. Oh, sorry. Okay, now I see that. Does the other 2 ADP come from outside of glycolysis? That is a fantastic question. I'm so happy that you asked that. The reason I think that's a fantastic question is because if you think about a cell and think about here's our eukaryotic cell, here's a mitochondria, here's the nucleus. Hey, my intent to draw the inner membrane of a mitochondria ended up looking like a lowercase m. How about that? This space out here, where I've drawn this X, go ahead and add to the chat what that space of your cell is called. Or what solution? Cytoplasm. The cytosol. Perfect. Okay, if you look at the space that a single cell occupies, there is a ton of space that constitutes your cytosol. And of course, this is a super simple model of a cell, but there's so many different processes that take place in that space in the cell. And of these processes, there are ADP generating processes. So almost any number of different like cell signaling or like if you think about, I'm going to draw a little protein here, receptor proteins. So a receptor that were to pick up a molecule like, and this is very intentionally poorly drawn, let's say caffeine. Caffeine goes into a receptor, it's perceived by a receptor, and it stimulates a response, and it makes ADP. Okay. That is one of many different processes that take place within the cytosol that generate ADP. So when you think about this ADP that's being generated from this, or these 2 ADP, those 2 ADP just kind of go off into the ether, into the cytosol. They're not necessarily the ones that get fed back here. Think about the way that I always try to think about it is within a cell, within the cytoplasm of a cell, there's really no shortage of ADP. The limiter would be ATP, a high energy compound. ADP by comparison is a relatively low energy compound. So I just want to make sure, so to answer your question, Does the other two ATP? Yes. And I would actually take it a step further and say all four of your ADP are probably coming from outside of glycolysis. That's a fantastic question. That's a great question because of you're thinking about the totality of it all. I like that. Um, but did I answer your question? Okay, cool. All right, so now if we're to jog ahead, here's that overview that I've looked at, that I've shown you before. And I know that this does appear like, oh yeah, ADP, magic. This ADP goes down here and that's how we make ATP. This is intended to simplify things. Now, we're going to revisit this figure later on after we've gone through our comprehensive overview. Today, what we're focusing on is the investment. Now, the first important detail of that investment is that glucose, our input molecule, can move in or out of a cell. So any endothelial cell, any muscle cell can take on glucose and it can release glucose. And you have regulatory hormones, glucagon and insulin. Your body or your pancreas produces insulin and glucagon and basically sends those out in response to how much glucose is in your bloodstream. If you have a low glucose concentration, well, then your pancreas is going to produce glucagon and it's going to say, hey, muscles, if you have any glucose, you need to release it because our overall bloodstream or our blood glucose level is low. Now, if there's a ton of blood glucose, well, then your pancreas is going to produce and secrete insulin so that all of your muscle cells get the signal of like, hey, there is so much sugar here. We've got to deal with it. We've got to process it. Now, that's the case with glucose. Glucose 6-phosphate or phosphorylated glucose or glucose phosphate, that's a different story because that gets trapped inside the cell. That's almost a way of determining, like, as long as there is a phosphorylated glucose, it's going to more than likely be used for some amount of energy. Now, using ATP to irreversibly convert glucose to glucose 6-phosphate allows regulation of glucose uptake. Okay, so here's our first step. One of the things that we talked about last week was linear glucose, which we've got right, left, right, and on our penultimate carbon, our our highest numbered chiral carbon, we've got a hydroxide group on the right-hand side. So we're talking about D-glucose. Now, between the two of these, so this can cyclize. And when it cyclizes, it's going to form either beta D-glucose or alpha D-glucose. Okay, so these two sugars, this and this, are anomers of one another. An anomeric pair is what they're also known as. Now, the more abundant and present one is beta D-glucose. That's kind of the more sterically favored form of this molecule is beta D-glucose. But beta D-glucose can interconvert through a process known as mutarotation to make alpha D-glucose. Now, that alpha D-glucose, that's the form of glucose that our enzyme, or our first enzyme in glycolysis, is going to use. So alpha D-glucose. So that's an important thing to remember, alpha D-glucose. Now that molecule is going to be phosphorylated with the help of an enzyme. Now an enzyme is a biological molecule that catalyzes a chemical reaction. So the enzyme hexokinase is our first enzyme in glycolysis. Now, hexokinase and glucokinase they're complicated because they're the same, but they're different. They carry out the same reaction, so they're the same. But glucokinase is found only in the liver, whereas hexokinase is basically in every single other cell. Now, what hexokinase and glucokinase are going to do is they're going to take adenosine triphosphate, ATP, and they are going to make glucose 6-phosphate. Now, I'm really happy that made the change to the curriculum and added this, because if you were having trouble with the numbering of your carbons in the cyclic form of glucose, I think that this is a really good way of figuring it out and reverse engineering it. If this is carbon number 6, which, when I provide you with the structure, you can see that, well, then what is this? That's 5, 4, 3, 2, and 1. Now, in the event that I gave you this structure and I didn't give you this name, well, then that's the sort of question you should anticipate and say, okay, well, I need to be able to decipher what the name of this molecule is. Okay, so I need to know which is my anomeric carbon. Well, that'd be that one. So that's 1, 2, 3, 4, 5, and 6. Now, the delta G of this reaction. So remember, our delta G is our measurement of favorability. This is the delta G naught prime. So thinking about it from a biochemist standard state is negative 16.7. What does that tell us? Does that tell us that our reaction is favorable or unfavorable? Favored. Now, it's highly favorable. It's spontaneous. Now, the tricky part about this is, well, this has kind of an equilibrium arrow, but we can see that it is favored in one direction. It's favored in the direction of making glucose 6-phosphate. So what does that say about the opposite reaction? Going from glucose 6-phosphate in ADP to make glucose in ATP, it's non-spontaneous. It's unfavorable. And we have a way of kind of quantifying the degree to which it's unfavorable. It's going to be positive 16.7. And that units for that would be kilojoules per mole. Now, this is a very tightly regulated and key regulatory step. We can look at that from a couple of different directions. One is that we have a negative delta G. Okay, so energetically, it's unfavorable. When you account for the concentrations of these different components within a cell. Next up is, this is a tightly regulated step because glucose 6-phosphate can't get out of the cell. Glucose 6-phosphate, for all intents and purposes, is trapped in that cell. Now, some of the different components, I'm happy that you asked that, Milo. That's a great question, and I'm happy that I have this slide for you right away. So magnesium is a divalent cation, Mg2+. Now, as a Mg2+, what it loves to do is it loves to attract electrons. So what magnesium is really doing here is it's helping stabilize the phosphate groups on our ATP molecule. Now, one of the things that I want you to be familiar with is if you look at ATP, it has a nitrogenous base, a sugar, and 3 phosphate groups. Now, if you look at it, and just imagine for a moment that this right here is our last phosphate group. It's the end of our molecule. It's the end of our ATP molecule. The naming for phosphates are alpha, beta, and gamma. That's a gamma symbol. Okay, so the gamma phosphate is the phosphate that is transferred. So the magnesium is what's stabilizing that. And so this brings up, what this addresses is the molecules and the atoms have to be positioned in a certain way in order for this reaction to take place. And what I mean by that is basically this divalent cation kind of holds those 2 phosphates. I always think about it like a, like a puppet master, a marionette. That magnesium ion is kind of holding these 2 phosphates in their proper position. Now, if we got rid of magnesium and replaced it with Mn2+, Manganese. Okay. Now, let's just think about the periodic table for a moment. Manganese and magnesium. Are they, and this isn't a trick question, are they in the exact same place on the periodic table? Magnesium is in group 2. Does anyone know which section of the periodic table manganese is found in? Add it to the chat if you do. I'll give you a little bit of a hint. It's a transition metal. Perfect. I didn't even have to give you a hint. It's a transition metal. Now, thinking about the periodic table and thinking about periodic trends, okay, let me just ask you this. Do you think the size of magnesium and manganese, do you think they're identical? No, they're not. Okay, so those two ions, a magnesium or a manganese ion, they occupy different space. What that means is if I replaced magnesium with manganese, this reaction's ability to take place is just going to plummet. It's going to bottom out because the structure where these two phosphate or where these two phosphate groups are is going to be altered. So magnesium and manganese, by being different, are going to alter this reaction's ability to take place. Now, I don't need you to push electrons. I know that this is showing you pushing electrons. I do want you to know that magnesium is a critical part, and here is what it does. I do want you to know that. Because this is something like tons of different processes and require ATP, and tons of different processes by proxy require either magnesium or calcium or manganese. They do require kind of a specific divalent cation. If you're taking it from this point forward, I would just go ahead and operate under the idea that, okay, I need ATP, I probably need magnesium. We'll get into some later on that require a different one, but I always just kind of think about if ATP is there, there's probably going to be magnesium. Okay, so this is what basically our reaction is. And if you think, I mean, whenever I think back to organic and I think about pushing electrons, this is like super simple mechanism. It's 2 arrows. I love it. It's not the, 14 different steps and 8 groups of electrons and 37 arrows that are being moved around. This is pretty cut and dry. Okay, so that's our first step, hexokinase. Glucokinase does the same thing. That's called an isozyme, but hexokinase is going to catalyze the phosphorylation of glucose to make glucose 6-phosphate. Now, next up, our next step, sorry, I just had to do the thing that I always do, and that is confirm that, yes, I am indeed recording, and I am. So the next step is taking glucose 6-phosphate right here and doing some simple rearranging to make fructose 6-phosphate. Now, this name gives you lots of information that you need, and that information is that, well, the enzyme's name is phosphoglucoisomerase. It's also known as phosphohexose isomerase, but I like to bring this one up because if you think back to Gen Chem, no. If you think back to organic, probably 1, you learn about isomers. The same atoms, same number and identity of atoms, but things are just positioned differently. And then you learn about constitutional, structural, all the different isomers under the sun. Here what we have is we have some simple rearrangement. We go from glucose 6-phosphate to fructose 6-phosphate. Now let's think about glucose versus fructose. What's the difference between them with respect to functional groups and classification? They're both hexoses. They both have 6 carbons. But what makes them different? And it might be helpful to visualize them or look at them as linear molecules. Glucose is an example. Okay. I was, Andrew, that's a great offering. I was looking for what Milo answered or suggested. Glucose is an aldose and fructose is a ketose. Spot on. What we're doing essentially is rearranging an aldose to make a ketose. This is a step that is completely reversible. This is a step much like the um, interconversion from alpha to beta sugar. That's a favorable reaction. It's kind of it's really not that much of a difference, but we have an aldose going to a ketose. So we've rearranged glucose to take on fructose. Now, this is kind of an interesting point, and I like to mention this just because if we jog back for a second and we look at this overall, if anyone's going to have I don't know, like a soda or something like that, or I mean, most anything, chances are they're going to have a little bit of high fructose corn syrup. High fructose corn syrup is a strong sweetener, and it kind of has a negative reputation. And the reason that it has, one of the reasons that it has a negative reputation is that, your body takes on all this fructose and basically it's more fructose than you're ready to deal with. Now, if you think about fructose versus glucose, well, glucose is going to go to make fructose 6-phosphate. If you were to ingest fructose, well, what your body is going to do is it's going to make fructose 6-phosphate. So on the surface, fructose versus glucose, molecularly, they're not that radically different. On the surface, if you ingest one molecule of glucose and one molecule of fructose, they're going to be metabolized slightly differently. But, the big problem with, or excuse me, one of the big problems with a high fructose corn syrup is not the molecule, but instead the concentrations and the volumes that your body takes on and how, the different organ systems will utilize them. So molecularly, they're super similar. And when you take on a small concentration of fructose 6-phosphate, your body processes it and makes, or when you take on fructose, your body makes fructose 6-phosphate, and that goes through glycolysis. But it's ultimately that concentration, the amount of it that you ingest. Okay, so then why glucose 6-phosphate to fructose 6-phosphate? Well, it provides an easy way for fructose to enter central metabolism. So we need fructose. Whenever you bite it into a strawberry, your body is going to get a lot of fructose. Now, the fourth step in this entire pathway is what's known as an aldolase. And we're going to talk about that on Wednesday. It's going to take your 6-carbon molecule and break it into 2 3-carbon molecules. So this, basically what we're doing is we're kind of priming our molecule to be used by aldolase. To get this fructose 6-phosphate, we need to make this, we need to make that so that the latter portion of this pathway can be used. Okay, now I'm going to skip over this mechanism. This is really just kind of if you want to take a look at it, you're more than welcome to. I'm not expecting you to know any of this mechanism because this is where we kind of pump the brakes. When you go on to take 4385, the metabolism portion, you're going to go over lots of the same content, but this is a slide that you'll dig into a little bit deeper and you'll be asked more about the mechanism because right now, this species right here, we haven't talked about what that could possibly be. We're speaking a little bit more generally about enzymes. So this would be a good resource to hold on to for when you take metabolism, maybe before the semester starts, you can breeze through this a little bit, make sure that you're in good shape there. Now, this brings us to our last of our 3 enzymes that we're taking on. So we have a total of 3 steps in our investment phase. The first step is hexokinase or glucokinase, where we take glucose to make glucose 6-phosphate. Then we have our isomerase step, phosphoglucose isomerase, where we make fructose 6-phosphate from glucose 6-phosphate. Our last step is our second big ATP investment step, in addition to being our second big investment step. So this is our second ATP is spent. 2. This is what is known as the first committed step. Now, this is a place where people get kind of mixed up and they get a little bit confused. Is this the first? And you can toggle through any of the PowerPoint slides, or here, I'll go back. I'm going to clear all the writings off of this so you can see it a little bit better. Okay. Phosphofructokinase. Take a moment and find that on this slide, and then answer this question. Is phosphofructokinase the first irreversible step? Perfect. It is not the first irreversible step. It is, however, the first committed step. Would anybody care to add to the chat or say it aloud? What do you think that difference is? Or is there a difference between those 2 terms? First committed versus first irreversible. I know I kind of asked a wordy question, or a question that has kind of a wordy response. I'm going to go forward. First irreversible step, just make sure glucose stays in, while the first committed step is the start of glycolysis. Milo, I love that answer. Okay, so we have to worry about gluconeogenesis. Okay, great. I like this. So you guys this is awesome. Those are fantastic responses. The first committed step, think about that as, what are we talking about? We're talking about glycolysis. The first committed step is the step where we're making a product. Our product is this right here. And the carbons of this molecule are going to proceed through glycolysis. What that means is that fructose 6-phosphate, well, that could go in, I'm just going to draw out some different directions. It could go this direction, not glycolysis. It could go this direction, not glycolysis. So it could go through different pathways, because if we think about metabolism, I mean, metabolism as a whole, is it just glycolysis? No, there's tons of different pathways. Fructose 6-phosphate can be used in 5 different pathways. Fructose 1,6-bisphosphate, however, that is one molecule that's been produced that's basically like, I'm going through the rest of glycolysis. That's all there is to it. So if we think about where products are going to be used, let's take glucose 6-phosphate. If you make a glucose 6-phosphate molecule, or let's imagine that you were just to take glucose 6-phosphate and magically inject it directly into a cell, not magically. If you were just to put glucose 6-phosphate into a cell, what are I'll treat it as a binary system. Put a thumbs up if into the chat if you think that glucose 6-phosphate could be used for something other than glycolysis. Yeah. Could it be used by glycolysis? Yes. Could it also be used by something other than glycolysis? Yes. Fructose 1,6-bisphosphate, by contrast, that molecule is so kind of destined towards glycolysis. That's why we refer to this step as the first committed step. We have committed to using the carbons of this product for glycolysis. Now, the second ATP is spent. It's our first committed step. I'm going to say that this is number 3, our second irreversible step. In addition to that, phosphofructokinase takes a ketose and phosphorylates a ketose, or it takes a phosphorylated ketose and makes a doubly phosphorylated ketose. So I want you to try to, whenever you look at something like this or a chart like this, I want you to be thinking about aldoses and ketoses. And specifically like phosphorylated aldose and phosphorylated ketoses. I think that's helpful because we are stopping right here today. On Wednesday, we're going to go the rest of the way. Now that seems a little bit like, oh man, there's a lot left. Yeah, there is. But from a understanding point or a conceptual understanding standpoint, I want you to look at the equilibrium arrows here. Reversible, 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 and then irreversible. From a regulatory standpoint, the investment phase is much more tightly regulated than the payoff phase, because the investment phase is where you do that heavy lifting and you have these irreversible steps, whereas the payoff phase, almost everything's completely reversible. So we will get into that on Wednesday, and that's going to kind of sum up and wrap up glycolysis. Now, I'm going to go ahead and stop my recording and stop my share, or I'll stop my recording first.